Hello, dear aspirants. Welcome back to the TEJAS series in GDC English classes. In this session, we're going to cover the MCQs related to the introduction of pharmacognosy part 3. So, in this introduction of pharmacognosy part 3 video or in this session, we are going to cover the topics like plant hormone, plant tissue culture, plant biotechnology and biosynthetic pathways so we are going to have the 30 MCQs from these four important topics from the part 3 of Introduction of Pharmacognosy. So those are nothing but plant hormones, plant tissue culture, plant biotechnology, and biosynthetic pathway. Okay. So let's start this MCQ session to boost up your preparation. Okay. Yes. In this series, let's start the first question. Okay, the first question in this series is the unsaturated 2-carbon containing molecules which involved in the organ abscission. Okay, option A, cytokinin, and option B, ethylene, and option C, gibberellic acid, and option D, auxin. So, let us go through the question once again. The unsaturated 2-carbon containing molecule which involve in the argon abscission. So you got to know that. So they were asking about two unsaturated carbon containing molecules. So you got to understand that it is a topic, it is a question coming from the topic called plant hormones. So in the plant hormones, what will be the two carbon containing molecule? So you can simply uh, choose the right answer by careful reading. What is a two carbon containing molecule here? Among we are having four options, right? Cytokinin, ethylene, gibberellic acid, auxin. Okay. So I think you got the answer. Yes. So option B is correct. Ethylene, two carbons, ethylene, carbon containing molecule which involves in the organ abscission. Okay. Ethylene. But the functions of ethylene is it is to promote the flowering and fruit ripening and also maturation and senescence. But organ abscission responds to the biotic and abiotic stress, inhibitor of root elongation. But whereas coming to the chemistry, it is an unsaturated 2-carbon molecule. Okay, so this is how the ethylene will make the fruit from unripened to the ripening fruit. So because the unripened fruit is consisting the acid, starch, chlorophyll, pectin, which is hard in nature, the larger molecules. And after spraying this ethylene, because it is in a gaseous form, okay, when you spray this ethylene, so it activates all the enzymes like kinase, amylase, hydrolase, pectinase, and hydrolysis. So then it will change the particular status of the fruit into the ripened. Okay. So then you will get that glossy, colorful, tasteful fruit. Okay. So then it makes a neutral sugar, less chlorophyll, less pectin, and increased fragrance to that of fruit. Okay, the function. So that's the first question in this today's stages series. Let us go for the second question. Which plant hormone helps in breaking the dormancy of plant? As we all know that every plant hormone, plant growth regulators and the promoters as well as inhibitors are having specific functions with respect to each type of hormone. Okay, because as our body also having different types of hormones having with a different functions. So that here the question is the which plant hormones helps in breaking the dormancy of plant? So among four options, one option is auxin, gibberellin, cytokinin, and ethylene. So what will be the right answer here? So which plant hormone was breaking the dormancy? Okay. So option B is correct. Gibberellin. So gibberellins are the plant growth promoters. Okay. So the gibberellin obtained from the Gibberella fusicorii. All the gibberellins are having a gibbon skeleton. Okay. So these are all the other facts we need to consider. So that what are the actions of gibberellin? So it can make a rapid expansion of plant cells and as well it can stimulation of seed germination. So this is how it germinates the seeds, breaking the dormancy, yes, and induction of flowering, marked increase in the marked increase in the stem elongation. So if you spray that ethylene, it can increase the stem elongation, increase in the size of leaf, and induction of parthenocarpy, and also leading to the seedless fruits. It's like how you can get, if you want to get the seedless fruits, so that you can spray, you can use this ethylene meanwhile in the cultivation and the processing of the fruits or maybe the plant. Okay, so these are the different functions of gibberellins. Okay, do remember the type of hormone it was and their functions because they are important. Okay, let us go for the third question. A typical auxin of plant which is used in growing tissue is, so which auxin? Because auxins are every plant hormone are synthesized naturally as well as they are prepared by means of synthetically. Natural hormones will be there. When the plant not able to produce or not able to synthesize the natural hormones inside the cellular structures, so then it requires the plant hormones from external resources. So that here they are asking about the question, what is a typical oxygen plant which is growing, used in a growing tissue? So option A, IAA, indole 3 acetic acid. Okay, NaA, 1 naphthalystic acid, and 2,4-dichlorophenoxyacetic acid. Okay, and 2,4,5-trichlorophenoxyacetic acid. So what will be the right answer here? So the option E is correct. That is indole 3 acetic acids. That is a natural oxygen. I know. Okay. So, but these auxins' main function is to promote the elongation of coleoptiles. That is, and this is a coleoptile. Okay. So, how these helpful? So, to promote the elongation of coleoptiles. So, coleoptile is a tissue will promote the germination, or it is something like a young shoot tip. So it will grows in a direction where the exact sun rays are, okay, falling on that particular. So let us see some of the examples for the natural and synthetic or synthetic auxins. So the plant itself had that IAA, IAN, okay, indole acetic acid is a principal auxin. Indole 3 acetonitrile is a IAN, that is a natural auxin. And 4 chloroindole 3 acetic acid, that is also natural oxygen synthesized inside the plant, and nothing but phenyl acetic acid. So these are natural oxygen. But what will be the synthetic oxygen? IBA, NOA, indole 3 butyric acid, 2 naphthyl oxyacetic acid, and alpha naphthyl acetic acid, 1 naphthyl histamide, 2,4-dichlorophenoxyacetic acid, 2,4,5-trichlorophenoxyacetic acid, 5 carboxymethyl N, and methyl dithiocarbamate. So do remember which one is a synthetic one and which one is a natural one. So try to remember these things, so the rest of will be fall under synthetic oxygens classification. Okay. So do remember these four things so that you will easily detect which one is synthetic and which one is a natural. Okay. So we'll go for the next question. A compound now increasingly used as a standard practice for enhancing the flow of rubber latex by spraying onto the scrapped bark of rubber tree, increasing the latex yields from 36 to 130%. Simple. Okay. So they were asking which compound is used to increase the production of latex from the rubber plant. Okay. So in order to increase the yield of some secondary metabolites, okay, some like resins or maybe any plant product, so we need to add some of the chemicals. So in that they were asking which compound is used to increase the latex secretion or latex yield in the rubber plant. Option A, prasinosteroids. Option B, abscisic acid. And option C, ethophone. And option D, kinetin. Okay, so what is the chemical or compound used to increase the rubber latex yield? Yes, option C is right. Ethophone is a hormone. Sorry, that's a chemical. Yes, ethophone is now increasingly used as a standard practice for enhancing the flow of rubber latex sprayed onto the scrapped box. So there is a tapping group of the rubber tree. It increases latex up to 36 to 130%. So usually this is a rubber tree. where you can observe in a different locations. But in order to increase generally the yield of this rubber tree is about 36%. But that 36% is not enough to produce a higher amount of rubber. So to make the wall to increase the yield of this particular rubber tree, so people are spraying this ethofon onto that scrapped bark. So exactly like this, so that the white colored latex, that rubber latex were increased or the secretion was increased. Okay, so this is about ethofon. Okay, yes. Next question is, plant cells are usually cultured on media having pH. So there are several requirements for the plant tissue culture technique, but from the plant tissue culture technique, the question is, plant cells are usually cultured on media having a pH. So what is a pH? 4 to 5, 5.5 to 5.7, 4.5 to 5, 3.5 to 4.5. So there are maybe some are acidic pH, some are basic, some are neutral, right? So there is no basic pH here. Neutral and acidic pH we can observe. But preferably 5.5 to 5.7 is required for a plant cell usually cultured on a media. Okay. Yes. So 5.5 nothing but more or less neutral to the basic. Sorry, neutral mostly. Yes. So these are the different requirements along with some ingredients and their function. So you can see the major gelling agent is agar. It is a growth medium and it requires some inorganic salts like macronutrients and micronutrients. In the macronutrients, sodium, potassium, okay, uh, and calcium, sulfur, magnesium, phosphorus, and micronutrients are ferrous, zinc, okay, manganese, copper, boron, and molybdenum. So these are all the micronutrients essential for tissue and cell growth. And whereas organic supplements like carbon source, so to give that required amount of carbon source, we need to add sucrose or glucose, because the sucrose or glucose are the carbohydrates. The carbohydrates are consisting of carbon, hydrogen, and oxygen in their functional group. Okay, so that the carbon is rich in carbohydrates. So the example of carbohydrates are sucrose and the glucose for the carbon source. Okay, the thing is, the question is for the plant cell pH is 5.5 to 5.7 here. Okay, so to maintain the acidic and basic in nature culture, and it requires some vitamins like thiamine, which is essential. That is nicotinic acid and pyridoxine. It improves cell growth. It also requires some of the amino acids like glycine. Okay, so glycine, which stimulates the cell growth and helps in the establishment of cell lines, and requires some of the plant growth regulators, because it is a plant tissue culture technique, is a new technique where we take the cell and we culture under aseptic conditions in controlled temperatures. Okay, in control conditions. Okay, so that the cell, because the after becoming a plant, the plant will create or it synthesizes its own plant hormones, but a single cell won't create or a single cell won't synthesize its own plant growth hormone. So for that reason, we need to supply them externally, so that growth regulators like auxins, cytokinins, ipperellins, oxalic acid need to be added, because they are having different functions to be in order to uh, get a good growth of the plant. So we need to have these auxins, cytokinins, ipperellins, and oxalic acid. So auxins uh, facilitate the cell division, root differentiation, induction and growth of callus, and cytokinins, it promotes the cell division, regeneration of shoots, and gibberellins enhance the callus growth, and abscisic acid induction of the embryogenesis. Finally, temperature will be 10 degrees centigrade is the best for growth. Do remember this slide particularly, so rest of the questions will be coming from this slide only. Okay, so most of the questions from the plant tissue culture, so these are the things they have asked for. Okay, yes, so next question in this series is, vitamin essential in tissue culture medium is. So just now I have said that about vitamins, enzymes, and hormones. Sorry, vitamins, hormones. Option A, pyridoxine, and option B, thiamine, and option C, nicotinic acid, and option uh, D-inositol. So what will be the right answer here? Undoubtedly, thiamine is a only okay vitamin required for the improve the cell growth. So here you can see vitamin and other like nicotinic acid, pyridoxine, and other things. Okay, so let us see the next question. Which of the following growth regulator induces the embryogenesis? Just now we have came across these things. Whether it is auxin or cytokinin or gibberellins or abscisic acid. These are all four different hormones, plant hormones. But which one of the following is induce embryogenesis? Option D, that is abscisic acid, will induce the embryogenesis. So you can see here abscisic acid induction of embryogenesis. Rest of the other plant hormones have different functions based on cell growth, cell division, sure regeneration, sure callus growth. Okay, do you remember the functions of plant hormones? Next question in the series: amino acid essential for the plant tissue culture. Just now we have chemicals. Option A glycine, B alanine, C thiamine, and D tryptophan. So these amino acids are synthesized in a pathway called ∑ acid pathway. Okay, so let us see which amino acid is essential in generally. Option A, glycine is a amino acid which is required for the essential for the plant tissue culture. Okay, so that is the eighth question. Next, we'll go for the next question in this series, and we'll have a brief explanation. Okay, in the nutrient media, in generally it requires inorganic salts, carbon source, growth regulators like auxin, cytokine, gibberellin, and ethylene abscisic acid, but vitamins like thiamine, but organic supplements like amino acids like glycine is required, and yeast extract, the pH is about 5.5 to 5.7. Okay, here the glycine is amino acids required for the cell growth. So, next question, ninth question in this series. Hormone which is responsible for the cell division in plant tissue culture. So, just now we have covered, okay, amino acid. Now they are asking about hormone, whether it is cytokinin or ethylene or auxin or gibberellin. If you properly read the question, you will get the answer. Cell division, cell division. Okay. So what will be the right answer here? Cytokinin, cell, division, kinins. Okay. Yes. So these are the media compositions in this. Uh, we require different things like we have covered up to now. So the growth regulators, cell division, hormones, cytokines. Now we can see, okay, uh, in the previous years of GPAT examination as well as GATE examinations also, these questions are appeared. So about the gibberellic acid, what are the natural uh, gibberellic acid or natural gibberellin? They were asked. Okay, so GA1 to GA2, GA3, and the function is food elongation, and the gate 6, sorry, gate 2006 and GPAT 2011 also we got question from cytokinins, and in the year of gate 2006 and 2012 GPAT and 2014 GPAT examination also we got a question from abscisic acid, especially from plant growth regulators. So it's an important topic, so do remember. Yes, and lastly, organic supplements like malt extract, yeast, coconut milk are used to enhance the growth rate of cell and biomass. Okay? Yes. 10th question in this series, the essential ingredient in the general preparation of plant tissue culture media is option A, auxin or naphthalenic acid, NA. Option B, sucrose or glucose. Option C, gibberellin Z1 and gibberellin G2 and pyridoxine here. Which one is the right option here? Let us check the right answer. Option B is correct. Okay. So the carbon source, sucrose and glucose, the source is energy, essential ingredient to general preparation of plant tissue culture media. Okay. So this is a very important slide. Try to capture this one, otherwise try to note it down. These functions and the important ingredients in the nutrient media are very important with respect to plant tissue culture. Next, 11th question in this series, in plant tissue culture, which of the following macronutrient used to maintain a vital function in the culture media? In the organic supplements, okay. Uh, in the plant tissue culture media requirements, there are macro and micronutrients are there. But in the macro, so what are the macronutrients used to maintain the vital function culture media? Either boron or copper or iron or potassium. So what is a macronutrient here? So potassium is a macronutrient. So you can see here macronutrient. It's an inorganic salt. Okay, potassium, phosphorus, calcium, carbon, nitrogen, sulfur, and magnesium are the here they are the inorganic salts and especially macronutrients. Okay, carbon source like sucrose, glucose, growth regulators like auxins and everything. Vitamins like thiamine is important, amino acid like glycine is important. Okay, do remember because why we are keep on explaining again and again because they are very important. 12th question in this TEJAS series part 3, that is the most effective method of producing virus-free plant is. So now we have entered into the types of culture techniques. One is root culture, hairy root culture, meristem tip culture, floriculture, somatic embryogenesis. Because every method of culture technique has unique application. By root culture, we can get a different application. By meristem tip culture, we can get different application. By floriculture and somatic embryogenesis. So, but about this question, by following which method can create or produce a virus-free plant? We will see the right answer, that is meristem tip culture. So this is the diagram how the meristem tip culture was carried. So you can see the propagated from apical shoot tip and used as a source of sterile shoot culture, which are further regenerated into virus plant. Yes. So this is a shoot tip. In generally the plant we have two parts, there is root and shoot. In the shoot, we get a shoot tip. So from this fruits shoot tip is sterilized and is again further cultured, like the surface of this shoot tip is sterilized by using sodium hypochlorite. The sterilized shoot tip is washed with distilled water and then it is taken out and leave it for few minutes and take the scalpel, okay, and make the incisions and take the apical tip and to that apical tip transfer into the nutrient media or the Murashi's and Skook's medium, which is containing all the nutrients and everything. So incubate it for 16 hours at 25 degrees centigrade under the fluorescent light so that you will get the root development. So that root is transferred to the pot. Okay. So because we have sterilized here, surface sterilization has done, and also we have taken the shoot tip, and because we have sterilized with sodium hypochlorite and we have placed it in a murasage and a Skook's medium and we have incubated for 16 hours, so that because of this procedure we can regenerate the plant into virus free plant because we have sterilized, surface sterilization has done with sodium hypochlorite. Clear? So that is the main application of the meristem culture so that we can get a virus free plants. Next question in this series. In callus culture, roots can be induced by the supply of. In callus culture, roots can be induced by the supply of. Okay. Option A, auxin and no cytokinin, only auxin. High concentration of auxin and low concentration of cytokinin. And C, higher concentration of cytokinin and lower concentration of auxin. and D, auxin and cytokinin in equal proportions. So what will be the option? So in culture, which one of the following proportions are used? So we'll see the right answer, that is option B is right. High concentration of auxin and low concentration of cytokinin required. So in a callus culture, it is an unspecialized, unorganized, growing and dividing mass of cell. You can see the flow chart. You can see it, you can take an explant. So the explant is placed in nutrient media. So undifferentiated cell mass, that is callus. So from that embryoid we can isolate the embryo, and there is a from that embryo. So germination has done plantlet. So again it is as plant. But to it produced when explants are cultured on the appropriate solid medium with both an high oxygen concentration and low cytokine concentration in correct condition. So 2,4-D are commonly used. Okay. Yes, during callus formation, there is some degree of de-differentiation both in morphology and metabolism, resulting in the loss the ability to photosynthesis. Yes, that's one of the drawback of this callus culture technique. Okay, so simply here the answer is for the callus culture for the root incision, we apply high auxin and low cytokine. That's it. So as we all know that every day we are looking at this poster, looking at this image or this slide. So as you all know that ZDC providing the pharmacist crash course, which was already started from 18th August. Okay, so uh, to make your preparation very uh, ease and to boost up your preparation, try to enroll and get registered in this course so that you can get lot of key features like live classes, recording lectures, test series, and previous MCQs and D notes. Okay, so I think the offer is going on. Hurry up, grab the chance, and boost up your preparation. Okay, yes. Next question in the series. The 14th question of this series is polyploid is defined as it is. Now we have entered the plant biotechnology topic. Okay, so addition of one chromosome or multiplication of entire chromosome set, submicroscopic change in DNA material, cross structural change. So that is what is polyploid? Option B is correct. Poly means many, ploidy means set of chromosomes. You can see the specific number of chromosomes is a characteristic of each species and it's called as genome. Yes, a set of chromosome is known as genome. And the when organism contains more organisms, two genomes is called as polyploidy. More than two sets of chromosomes is known as chromosome sets is known as polyploidy. The type of polyploidy which genome contains whole set of number of euploidy. Then thus euploidy includes monoploidy. If one set of chromosomes is there, monoploidy, two sets diploidy, and more than two sets polyploidy. And when the organism contains more than two genomes, it's called as polyploidy. But here, from the polyploidy, we can expect some other type questions like, what are the agents we can? What is a agent which can cause a polyploidy? What is a physical agent and what is a chemical agent? In generally, the polyploidies can be done manually or maybe experimentally. So in order to create that polyploidy, we use a physical agents like X-ray, centrifugation, and temperature soaks. If you do these things, the chromosomes sets will be multiplied. So in order to create that more number of chromosomes by using chemical agents, we can rely on colchicine, veratrin, sulfanilamide, hexachlorocyclohexane, and mercury chloride. So these are very important. But among all, colchicine alkaloid is the best known for the chemical to cause a polyploidy. Among all, colchicine is a good example for C chemical agent. C colchicine chemical agent which cause a polyploidy is colchicine. Do remember that. So these are all okay representing the polyploidy. Three number of sets are there. Okay. Next question, the 15th question. Callus is in the plant tissue culture where we can get so many new terms. Callus, explants, okay, colonies, embryogenesis, embryos. Okay, so let us see what is a callus. An organized mass of cells, an unorganized mass of cells, an unorganized mass of nucleus, and organized mass of nucleus. So what is the answer here? Callus is a, I think so. The option B is right. Undifferentiated cell mass. Okay. Mass of cells. So you can see these are the image, real images of what we can see the callus. The callus is an unorganized and undifferentiated mass of cells referred to as a callus. So plant tissue culture is a suitable medium divided to form a callus. Okay. So after taking an explant, after taking cell, like you know that explant, so it should form a callus. That callus regeneration to that cell walls, and again the entire new plant is developed. So callus is a growing mass of unorganized plant parenchyma cells. Callus is a undifferentiated mass of plant as well as it is uh, unorganized plant parenchyma cells. Parenchyma cells. Okay. So half of the MCQ is completed. Let us cover the rest of the uh, MCQs. So keep that spirit. Stay focused. Polyploidy is induced through option A, irradiation, mutagenic chemicals, ethylene, and colchicine. What is the right option here? Just now we have came across through the polyploidy. So the answer is colchicine undoubtedly, because yes, now we have covered the polyploidy, the set of chromosomes known as haploid, diploid, triploid, tetraploid. Okay, and many organs have more than two sets of homologous chromosomes. That is called as a polyploidy, and it can have a various effects on a plants like Distalis, Mentha species, poppy. Okay, the plants containing tropical alkaloids like lobelia and etc. But we have seen all these things in the last slide. So colchicine is a chemical agent which can induce the polyploidy. Okay. Next 17th question in this series. Which one of the following is not a protoplast fusion agent? Not a protoplast fusion agent. Fusion agent. They were asking which is not a fusion agent. A, treatment with sodium nitrate, calcium at alkaline pH, polyethylene glycol, and colchicine. What is the right answer here? It is a colchicine, not a fusion agent. Okay. So in the protoplast, so but colchicine is important in polyploidy, but it is not a fusion agent in protoplast culture technique. So protoplast fusion is a physical method of fusion of somatic cells from a different plants to form hybrid. So this is an isolated protoplast from plant, sorry, potato, it's from tomato. Okay. So for the fusion of protoplast, it won't be used. Colchicine won't be used, but like sodium nitrate and calcium ions, polyethylene glycol, and electrofusion are the methods or the reagents or the agents used to or to create the fusion. Okay, so mixing of protoplasts of two different genomes and can be achieved by either spontaneous or induced fusion methods to create nothing but for hybridation. To create a hybrid, okay, so we need to use in order to hybrid the two different protoplast or somatic cells. We need to use sodium nitrate, calcium ions, PEG, and electrofusion. You remember that. 18th question of this series. Hairy root cultures of secondary metabolite production are induced by transforming plant cells with. Okay, simple question. Hairy root culture can be possible with A, virus, B, Agrobacterium tumefaciens, C, Bacillus thuringiensis, D, Agrobacterium rhizogenes. I think you know this answer. Undoubtedly, the last option, Agrobacterium rhizogenes, is the right option here. Okay, so for the hairy root culture, okay, we need to add the Agrobacterium rhizogenes infection. So a relatively new type of land tissue culture which consists of highly branched roots covered with a mass of tiny root hairs originated directly from the explant in response to Agrobacterium rhizogenes, used to produce a clean and good quantities of valuable secondary metabolites. So this is a flow chart. It's a flow chart. So maybe So by inducing the Agrobacterium rhizosinus directly to that plant, it can create the hairy roots. So rhizosecretion. So that again we need to scale up this particular the hairy root developed leaf part, or either directly from the seeds by getting a different explant. Also we can directly induce this Agrobacterium rhizosinus, so that induction of hairy roots, so that selection of hairy roots. So this is how the entire process is done. Okay. So from this selection of hairy roots, then cell differentiation is done, redifferentiation has done, regenerated shoots, so that the transformed plant has achieved. Okay. Yes, the 19th question in this series. Choose the correct key intermediate for the biosynthesis of C663 units, which serves as a precursor for the biosynthesis of amino acids. I think you got to know. We have entered into the MCQs which are related for the biosynthetic pathways. So they are asking that choose the correct key intermediate for the biosynthesis of C663 C3 units, which serves as a precursor for biosynthesis events. Simple here. In order to understand the right answer, or to get the right answer, simply rely on this one. So which of the following pathway get to prepare the amino acids? You got the answer. Okay. One option A cyclic acid, B pyruvic acid, C dehydrochemic acid, D malonic acid. Option A is right answer. From the cyclic acid also known as, from the cyclic acid, we do get aromatic amino acids as a final products, nothing but phenylalanine, tyrosine, tryptophan. Okay, so the phenyl propane derivatives like this, all tryptophan, phenylalanine, tyrosine are the C6 and C3 units. C6 and C3 units. Clear? Yes. One benzene ring, that is C6, and three carbon atoms at the para position. So next question in this series. Monoterpene and steroidal compounds can be biosynthesized by A. Shikmic acid pathway, B. Ornithine, C. Malonic pathway, both A and C in the D option. So what is the right option? Monoterpene and steroidal. Okay. Oid. What will be the steroidal compound can be synthesized in a pathway? In which pathway steroid compounds are synthesized? Yes, in the malonic acid pathway. It is also known as isoprenoid pathway, oid, steroid. Okay, so this is a pathway overview. So primary metabolites from the sugars in generally. Okay, if the from phosphoenol pyruvate, shikimic acid is carried. So from the shikimic acid, we do get aromatic amino acids like phenylalanine, phenylpropanoids. Okay, from these aromatic amino acids, we do get alkaloids, peptides, cephalosporins, penicillins. And from that phosphoenol pyruvate plus 𝘿𝐶𝑂A coenzyme A, from that malonyl CoA, it produces a fatty acids. That is a fatty acid pathway. There is malonate pathway. Okay, so from this malonate pathway, we do get anthraquinones. Okay, and the tetracycline, erythrocyanine, and from these fatty acids also we do get fats and waxes. But from this 𝘿𝐶oA to again two okay. Acetyl CoA compounds from the two acetyl CoA. So it acts as a precursor. From this, isoprenoids are produced. Those are isoprenoids or terpenoids. Okay. So from these terpenoids, the steroidal compounds can be synthesized. Clear? Yes. So this is how entire primary and secondary metabolic pathways has done. Yes, as you all know that not only for GPAT and as well as pharmacist, and for other courses like DI, State Pharmacist and Center Pharmacist, and Railway Pharmacist also, we GDC platform came up with another crash course, that is Railway Pharmacist crash course. New batch has already started from 18th August. Every day live classes will be held from 6 p.m. So, um, and also there is a free pharmacist marathon is also providing you if you are enroll this course. Okay. Yes. For more updates, uh, visit GDC English Classes app, GDC English Classes YouTube channel, as well as GDC Classes Hindi. Okay. The 21st question. Quinol and alkaloids are biosynthesized via which one of the following pathway? Quinol and alkaloids. Either by ∑ acid, from ∑ acid as a tyrosine precursor, as a ∑ acid tryptophan precursor, ∑ acid cathinone, ∑ acid phenylalanine. What is the option here? Option B, ∑ acid and tryptophan is the right answer. You can see the explanation here. So you need to know quinol and alkaloids can be produced by tryptophan. It is a very important table. You need to remember that because these are all the amino acid precursors, and these are the example of the secondary metabolites that are synthesized by these precursors, and these are the classification. Okay, so please go through this slide. So tropane alkaloids, pyridine, pipyridine, tetrahydroisoquinone, benzyl tetrahydroisoquinone, alkyl element, terpenoidal tetrahydroisoquinone, indole, pyroindole, quinoline, quinazoline, imidole, amine. So these are all the examples, and these are the precursors. Where do we get these type of examples? Okay, yes. And also this pathway can also give you a brief idea regarding the ∑ acid pathway and their precursors. From the ∑ acid, the crosmic acid is formed. From that crosmic acid, anthranic acid is formed. So from that anthranic acid, tryptophan is synthesized. From the tryptophan, we can get indole phytogenes, and indole and quinone alkaloids are produced. Okay, so from phenylalanine, the rest of the secondary metabolites are synthesized from tyrosine. These are the things. Do remember this flow chart so that will be helpful for you. 22nd question in this series, that is biosynthesis of 𝘿𝙄𝙎𝘾 follows the which pathway? Either ∑ acid pathway, mavolinate or mavulinic acid pathway, both A and B, or estate pathway. So what is the answer here? Option C is correct. Both A and B can be synthesized by 𝘿𝙄𝙎𝘾. Okay, so 𝘿𝙄𝙎𝘾 from the both biosynthesis of 𝘿𝙄𝙎𝘾 reveals that ring A is derived from the ∑ acid, while the mavonic acid incorporated into the ring C. So ring C is synthesized from mavonic acid, and ring A is synthesized from the ∑ acid pathway. So it can be synthesized by both pathways, ∑ as well as mavonic acid pathway. 23rd question in this ∑ acid in the series is, in ∑ acid pathway, crocimate mutase converts a crocimate 2 or crocimate 2. So in the ∑ acid pathway, the crocimate mutase enzyme converts the crocimic acid into either carotenoids, phytol, frephinide or frepinic acid, and gutta. What is the answer here? We'll see the right answer, frephinate. So here you can see that frepinic acid, okay, from this ∑ acid, crocimic acid synthesized. From this crocimic acid, the crocimate mutase can synthesize frepinic acid. From this phenyl pyruvic acid and uh, p-hydroxyphenyl pyruvic acid synthesized, so that rest of the phenylalanine tyrosine has synthesized. 24th question in this series, atropine biosynthesis involves which of the following amino acid? Ornithine, tyrosine, dopamine, and tryptophan. Atropine biosynthesis involves in which of the following amino acid? Let us see the answer. Ornithine. You can see from the ornithine, tropane alkaloids, example atropine. That's the reason I have said. Do remember the precursor and do remove the examples which compound has synthesized from which precursor. Okay. Yes. 25th question. Colchicine is synthesized from either tyrosine, tryptophan, ornithine, or leucine. Sorry. Tell me the right answer here. Yes, we will check. Tyrosine. as you can see, uh, from the tyrosine, this colchicine has produced. Okay, from phenylalanine, epinephrine, and mescaline has synthesized. But this colchicine plant or the colchicine biogenetically derived from tyrosine. Okay, and we need to cover the other important bits like um, colchicine is a chemical inducing agent of polyploidy, and also it is not a fusion agent in the plant tissue culture technique. So that we need to revise. Okay, yes. Now we'll see the next question. 26th question of this series. Which one of the following alkaloids derived from the tyrosine? Hemitin, chelidonine, lobelin, stachytrine. Okay, stachytrine. So what is a alkaloid produced from the tyrosine? What is the right answer here? We need to check. Hemitin, yes. From the tyrosine, hemitin has synthesized. You can see from ornithine, nicotine, cocaine, scopolamine is synthesized. From lysine, lupin, anabasine, and you can see isopelatyranine has synthesized. And from the tryptophan, uh, ornithine, tryptomine, strychnine, reserpine, vincoside has synthesized. Whereas from phenylalanine, epinephrine, mescaline, triboic acid has synthesized. From tyrosine, papaverine, thebaine, codeine, morphine, as well as dihydroxyphenylalanine and dopamine. From the dopamine, epinephrine and colchicine has synthesized. So, amitin is synthesized by the tyrosine. This is the question. This is the answer. And 27th question of this stage series. Precursor of anabasine is from lysine or leucine, phenylalanine or tryptophan. So what is the answer here? Let us check. Lysine is a precursor for anabasine. You can just now we have discussed anabasine. Here you can have the anabasine. Okay, lupin, anabasine, and isopelatyrin are the compounds inside from the lysine as a precursor. Okay. Yes, 28th question from the series. Anthralinic acid is an intermediate precursor in the formation of tyrosine, tryptophan, ornithine, and methionine. So what is the right answer here? Yes, you need to post on the chat box. Okay, don't look at that. Just to respond, post your answers so that we know you're watching. Okay, yes. So we'll see the right answer. Tryptophan. From the tryptophan, anthralic acid is intermediate precursor in the formation of tryptophan. Okay, so here you can see the anthralic acid. You can see the anthralic acid here. Okay, so to prepare to synthesize the tryptophan, this amino acid pathway or shikmic acid pathway do prepare or do synthesize the anthralinic acid before the tryptophan. Okay, that's the 28th question. And we're almost end to this session. Which one of the following alkaloid derived from the lysine? Either it is amitin, chelidonine, lobelin, stachytrine. Stachytrine. Okay. So post your answers quickly. Okay. So I'll go through with the slides. You need to post your answers. Meanwhile, watching the video. Okay. Yes. Option C, lobelin is a alkaloid derived from the lysine. Here it is explanation. Okay. Lobelin from lysine. Okay. It is a piperidine alkaloid. Lysine, lobelin, piperidine. Lysine, lobelin, piperidine. We're almost end. The last question of this today's stage series question is sesquiterpenes are biosynthesis from in plants. From which compound? In plants. Farnesyl pyrophosphate, geranyl farnesyl pyrophosphate, terpenes, and degraded products of terpenes. Sesquiterpenes. So it is an pathway. Okay. Mavronate pathway. So the option is right, farnesyl pyrophosphate. where you can see the isoprenoid pathway. These are all the units from geranyl pyrophosphate, monoterpenes are synthesized, and from the geranyl pyrophosphate, farnesyl pyrophosphate is synthesized. So from the farnesyl pyrophosphate, sesquiterpenes is synthesized. Again from this farnesyl pyrophosphate, isopentenyl pyrophosphate is synthesized. From that isopentenyl pyrophosphate, we do get squalene, triterpenes, and steroids. From this again, diterpene has synthesized. From the diterpene, we do get gibberellin, phytol, tetraterpenes, and polyterpenes, and rubber and good. Yes, this is for today TEJAS series. For more updates, follow GD's English classes and do follow all the videos and do watch all the videos. Those will boost up your preparation. Definitely do watch all the remaining videos in the GDC English classes YouTube channel. Thank you. We'll see in the next class again with another set of 30 MCQs from Pharmacognosy. Thank you. Have a happy learning.